All right, so thanks everyone for coming, and thanks RMIT for the invitation to speak. It's really a pleasure, and it's exciting to see all these different applications of network science. So I'm going to talk about wildlife epidemiology and the way that I have used network science to make advances on work in different disease systems. And the talk has two halves. The first half is about a disease system in Kazakhstan that I worked on as a postdoc, and that's what got me into network science in the first place. And it's a nice part of the talk. It's kind of like a bit of a slideshow of Kazakhstan, bit of wildlife and so on. Then the second half of the talk is about my work here in Australia, where in collaboration with CSIRO, I've been working on different disease systems here in Australia. And in that second half, I just want to really acknowledge Dr. Jess Hopf, who is a postdoctoral researcher who worked with me the last few years, and so she deserves quite a lot of the credits. All right, so let's talk about Kazakhstan. So it's a plague system. This is bubonic plague, Yersinia pestis, the Black Death. Everybody knows what the plague is, right? And what you're seeing here is a great gerbil. It's a sizable rodent that carries the plague bacteria, and they have outbreaks of plague in Kazakhstan due to this innocent-looking great gerbil. All right, so these gerbils make burrow systems in the desert, and you can see one in front of you. They're quite big. They live in family groups, and they construct these complex burrow systems that protect them from predators and heat and cold, and this is where they live, and they defend these burrow systems to the death. They're really important. All right, so our data consists of surveillance that was going on in Kazakhstan since the end of World War II, right up until the independence of Kazakhstan from, the good old USSR. And they would collect these, they would trap these gerbils in these wooden boxes and stuff them into a sock. So that's a gerbil you can see that's been stuffed into a sock, and the reason is because the pressure on the fur makes the fleas come out. So if you look closely, you can see the fleas coming off the rodent. So they collect these fleas, they collect some organs and blood of the gerbils as well. And they try and grow the Yersinia pestis bacteria. And they have been doing this forever. So they've been doing it for 40, 50 years. As I said, from World War II, the end of World War II. And so what, hang on. So when they do discover plague, they do go and control it. But what I really want to talk about is the time series. But the slide here is, there's a truck with a dirty big pump, and they're pumping insecticide through the burrow systems of these animals. And the insecticides kill the fleas, right? The last thing you want to do is kill the rodents, because then the fleas just jump off and find something else to suck blood from. So they pump insecticide and they get rid of the fleas and they prevent outbreaks. So this whole surveillance program gives us an amazing data set to work with in terms of epidemics of plague in this host. All right, so let's go through this a little bit carefully. So there's two vertical axes, right? There's a time series of occupancy, which is an index of abundance. How many gerbils are there? And then on the other axis, you have prevalence in rodents. So this is indicating the presence of plague. So basically, this histogram tells you when plague was around, and the line the line and the points is the abundance of the great gerbil. And in the Northern Hemisphere, rodent populations always cycle. They have these multi-year cycles, and that's exactly what you see here. There's a sort of 10-year cycle, 7 to 10-year cycle. So there are periods, 4 or 5 years, where there's gerbils everywhere. And if you live in the country at the moment in Australia, you can relate, because we've got mouse plagues happening in the farming country of Victoria and New South Wales. So, you know, house mice don't cycle in Australia, but they have, we have plagues of house mice. All right, so these gerbils are cycling, and you can see, you can pick up a bit of a pattern. I'll show you another site where if you get a peak in abundance of the gerbils, you get an outbreak of plague. And you can see this in this time series, but what happened was fascinating when we pulled these data together and just plotted presence or absence of the bacteria. That's these open circles and filled in circles. And a 0 means plague wasn't detected, and a 1 means plague was detected. And then on the vertical axis, the horizontal axis, you've got this measure of abundance that I talked about, which is occupancy, and that's an index of abundance, and that describes how the population is going up and down all of the time. And what we see is that there is this threshold behavior in the appearance of plague. If the abundance of gerbils is very low, then you never see plague, you don't detect plague. But once you get to about you know, a particular abundance, then you start to see outbreaks of plague. And this is actually a very strong result. It got published in Science in 2004. And that's because thresholds for disease in general are expected by the theory, but they're very rarely observed. And so science was quite interested in this. A lot of the policy around wildlife disease, particularly around culling, depends on this idea that if you have a low abundance, then transmission of disease is more difficult. The contact rates aren't as high, the population is not as connected. And so, you know, part of the justification for killing things, and in Australia too, is this theoretical idea that low abundance means less disease. All right. Now, where does this threshold come from? And we finally get to talking about network science, and it's a percolation threshold. I don't want to use up all of my time talking about this system in Kazakhstan, but what we realized is it's an extremely spatial system. And so this is on Google Maps, and what you're seeing is, you know, you can see the vehicle track running from left to right, and you're also seeing these burrow systems of these gerbils shining out. So what happens is the vegetation disappears from the top of the burrow system, and you just get very sandy soil, and that reflects the sunlight relative to the surrounding area. And so what we realized is that the bacteria was spreading across the landscape in a percolating way, if you like. And so this comes back to graph theory. And this is just bond percolation on the square lattice, if you've ever seen this. And if you vary the probability of a bond forming between two neighboring nodes in this lattice, then as the probability increases, you get this really strong dynamic that a giant cluster, they talk about a giant cluster appearing at a particular probability. And that critical probability is a half in this case. So this is percolation theory. And you know, the similarity between what we were seeing in the plague data in the gerbils and this percolation behavior, where you have a critical threshold above which you form a giant or infinite cluster, has parallels, right? So it was really satisfying when we put together the spatial scales of the plague system, and we used a network model to look at the spread of plague bacteria and this threshold behavior. So what's really neat is that the scale of observation here is 10 kilometers by 10 kilometers. So the Kazakh authorities would divide the landscape up into small squares like this, and they would visit squares. And so the scale of observation is about this 10 kilometer by 10 kilometer area. And then you have the scale, the density of the burrow systems in the desert. And thirdly, you have the scale of movements of fleas and gerbils. And you need movements of gerbils and fleas to get transmission of bacteria between burrow systems. And we got this really nice result. And I was lucky enough to get this published in Nature as well. And if you're into graph theory, if you're into percolation theory, this is a really nice example to know about as a real life example of percolation. It's such a key idea in graph theory. It's great to see it have a real application. All right, so what I want to draw your attention to here, and we're going to take a step back from Kazakhstan and look at some big picture stuff, is that the threshold coincides with R0 is 1.5. So what I wanted to talk about was this sort of continuum from very, very spatial disease systems where a lattice-like graph is a good representation of the host population, to on the other side, which is more like us as humans. So if we think about COVID-19, I know you're probably sick of thinking about COVID-19, but humans living their normal lives are the complete opposite of gerbils living in burrow systems in Kazakhstan. Humans are flying around, we don't have a fixed location, our contacts can be random, and very unpredictable. And in the case of humans in COVID-19, you get exponential growth. And that's why they talk about flattening the curve. That's why public health authorities, get very worried. And, increasingly we're jumping on outbreaks earlier and earlier because of this exponential growth, right? It can so easily get out of hand. All right, so I find this a really interesting way to think about things, because you start to make more sense of curfews and limits on travel. The kinds of things we're facing during lockdowns restrict our movements so that we actually become more like the gerbils in Kazakhstan. You can't go more than 5 kilometers from your house. And, this pushes things to be more spatial. And, the difference between a spatial system and a non-spatial system is profound. So in the case of a spatial system, you do not get exponential growth, you get linear growth. Everything slows down. So, when you go into lockdown and you're not allowed to go further than 5 kilometers from your home, you can understand better, right? You can understand better what the advantages of this is. All right. So let's move on to my work in Australia and this really neat application of community detection, actually. So in collaboration with CSIRO, the Australian government asked us to consider the release of a cyprinid herpes virus if you've heard of Carpageddon, it was in the newspapers 2 or 3 years ago. It's very controversial. A lot of people don't think we should do it. A lot of people think we should do it. You know, Australia has had success with rabbit control in the past by releasing viruses that are novel to Australia. The hope is to, you know, to replicate that for the common carp, which is an invasive species in Australia, causes all sorts of problems. All right, so together with CSIRO, we developed a disease model for the spread of this herpes virus in Australian carp populations. And, you know, the stakeholders were extremely concerned to capture as much sort of Australia-specific detail as we could. And so what you're looking here is a part of New South Wales. So if you look at the top graph here, on the top half of the slide, each of those little yellow dots represents a piece of river or a water body, such as a lake or a dam. You know? And all of these water bodies have carp in them, and our task was to model the spread of a herpes virus through these carp populations. You know, in particular locations of Australia. So we had flow data, we had water temperature data, we had carp abundance data. And, you know, in the end, we built a monster of a model that was very complicated. But I want to talk to you about a step in this modeling process where we used community detection. And what we did was we said to ourselves, oh my God, we can't possibly model every one of these points, right? There are hundreds and probably more than 1,000 of them. And to manage the scale of the problem, we used a random walk or a random swim algorithm to detect communities of nodes that were well connected. So from the perspective of the fish, you know, swimming around this catchment, what constitutes a community for that fish. And so you sort of so the bottom half of this slide describes what happened when we did this random walk analysis. And it was really nice. It worked beautifully. So here's a couple, just want to show you a couple more examples, and apologies for the quality of the slides here. This is another one. This is a glenalg. And so you know, we went from thousands of nodes to you know, a dozen or more communities of carp. And that just made the whole disease modeling task much more doable, much more feasible. I wanted to show you another one. So this is the most interesting one. This is in Victoria. This is the Mid-Murray. And this is interesting because even though it's a river system, it has a cycle in it. So you can see a cycle in the top part of that graph. It sort of looks like a seahorse. It's kind of a beautiful picture of a network. Almost a seahorse, right? So we were particularly happy with this one. Good. Okay, so last one. This is another section of this is the Lower Murray, I think. And it just worked really well. It's worked so well that what we want to do is to try the same sort of idea for other problematic invasive species in Australia. So this is my last slide. How am I going for time, Arathi? Do you want me to shut up quickly?
You're doing good, Stephen. You're doing good.
Okay. Yeah. So I have a few more minutes to talk. Okay. All right. So yeah, feral pigs. This is my next project. Again, it's in collaboration with CSIRO. The major concern is again disease. So we're worried about African swine fever coming into North Australia. And, you know, if it gets into the feral pigs, we're never going to get it out again. So we'd really like to be able to, we'd really like to be more prepared for this happening. And we'd also like to manage our feral pigs better. They're continuing to spread. I don't know how many of you know about the Otways in Victoria. This is a beautiful national park of coastal, you know, coastal mountains, I guess you call them. Not quite mountains, but it's really beautiful. And the feral pigs have gotten in there, and I hike there, so I'm particularly upset that these feral pigs continue to, you know, damage our national parks. All right, but do you know feral pigs are a lot like fish? They tend to hang around near rivers. They're highly dependent on water. So pigs and fish, you know, actually follow the catchments. They follow wherever there's water, that's where you'll also find pigs. So the idea is that we want to map these national parks and try to work out the connections between feral pig populations, and, you know, whether we can divide and conquer. I mean, that's the real trick here. At the moment, the management of feral pigs is terrible. We just go out and shoot them, basically. And then they come back, and then we go out and shoot them again. It's not working very well at all. But we need to get a handle on the scale of management. What is the spatial structure of the pig population that will allow us to divide and conquer and manage these pig populations better? And that's where I hope to use network science to try to work out the connectivity between the pigs. It's exciting because they're also collecting samples and using genetics to try to work out the relatedness of different pigs in Australia and the gene flow between different populations of pigs. And so that, together with this sort of network science ideas around random walk analyses and representing the habitats of the pigs as a network, I think it will be great. And I look forward, maybe we'll do this again in a year, and I can present some results at that point. All right, so thanks for listening. I'll stop there.